Warm greetings to you all. My name is Tony Fauci, and I am the director of the National Institute of Allergy and Infectious Diseases at the NIH. It gives me great pleasure to introduce one of today's speakers at the InterSystems Virtual Global Summit, Dr. Bruce Walker. When I first met Bruce 35 years ago, he was still in training as a physician scientist and had just described a key immune response that allows the body to fight back against HIV. Ever since, he has been a highly valued global leader in the field of HIV and immunology, committed to collaborative science to solve pressing global problems. 10 years ago, he took his vision to a new level with the creation of the Reagan Institute of Massachusetts General Hospital, MIT, and Harvard, with generous funding from Terry and Susan Reagan. The Reagan Institute's mission is to harness the immune system to prevent and cure human disease by bringing scientists, engineers, and clinicians together to better understand and make research progress against the most formidable infectious diseases we face today. Reagan members have made great strides in developing vaccines to prevent Ebola, Zika, and HIV, with a candidate HIV vaccine partially funded by Terry and Susan now in phase 3 trials. And in keeping with the theme of InterSystems Virtual Global Summit, Adaptability in a Changing Environment, last January, even before COVID-19 became a pandemic, Reagan scientists, under the leadership of one of its founding members, Dan Baruch, rapidly adapted their HIV vaccine platform to target SARS coronavirus 2, a new and deadly coronavirus. Their vaccine candidate, developed with partners Beth Israel Deaconess Medical Center, Johnson & Johnson, and the NIH, is now in phase 3 human clinical trials. Ultimately, science will be front and center in ending the COVID-19 pandemic, and it will do so through the application of collaborative interdisciplinary research and through the ability to rapidly adapt to new challenges and turn them into opportunities, hallmarks of Reagan Institute under Dr. Walker's leadership. I would like to introduce to you now the director of the Reagan Institute and my good friend and valued colleague, Dr. Bruce Walker.
Thanks very much, Tony, and thanks very much to InterSystems for inviting me to speak to you today about the quest for a COVID vaccine and the need for agility in the life sciences. In 1981, I had just finished medical school and had started my internship at Mass General, was working in the emergency room when a patient came in with a disease that I had never seen before. In fact, the doctors there had also never seen it before. The patient rapidly died with diseases that up until then I had been taught never occurred in 19-year-old individuals. This person had Kaposi sarcoma and pneumocystis carinii pneumonia both at the same time, and it turned out that physicians across the country were seeing these new cases. That was only clear after the fact. And by 1981, there was the first report of this new disease. We didn't have a treatment for it. We didn't know what was causing it. And it took another 3 years for HIV to be identified as the cause. And it took 4 years from then to come up with a diagnostic test to be able to tell just how widespread that pandemic was. I had come to medical school intending to become a family physician, and it was really seeing those patients that made me realize we as physicians, if we didn't learn from patients, we would watch a lot of our patients die. And that prompted me to become not just a physician, but also to try and learn from my patients as a scientist to try to understand how the body fights back against this new disease and why it usually loses that battle. As I was continuing to see patients and trying to understand that, something really remarkable happened when I saw a patient in 1994 in the clinic. This person came in, and these are my clinic notes from that time, saying that he had been infected in 1978, right before Thanksgiving, with symptoms that were hard to describe. It was a severe flu-like illness. He felt like he was frozen in the mud. They thought initially that maybe this was a cerebral hemorrhage, a bleed into his brain because it was a hemophiliac, but in fact they never made a diagnosis. And it was in 1985 when a diagnostic test for HIV was available that they were able to go back to frozen specimens and realize that this person actually had been infected in 1978 with HIV, probably one of the first patients to become infected. When I saw this patient and realized that this person had been infected for 15 years, first I said, oh, well, I don't think you can possibly be infected because everybody dies. But as we looked into it, it was clear he was infected, and yet he was doing something extraordinary. His own immune system was controlling the virus without the need for medications. In fact, medications to treat HIV effectively were not yet available. That led us to pivot immediately to start to try and find more of these individuals. We established the International HIV Controllers Consortium, which was taking a completely new approach to trying to identify patients. By going to physicians really around the world, looking for other individuals that controlled the virus spontaneously without the need for medications. At the same time that we were realizing from these outliers that we could learn so much about how their immune systems might be dealing with it, we also realized there was another outlier group, which was babies being born with HIV infection. This is a picture I took from the clinic we were working out of in South Africa, where we were testing pregnant women and realized that 50% of them were HIV infected. No medications available at that time, and people living in really terrible poverty. We realized that there was so much that could be learned by studying this new pandemic at the heart of the epidemic, at the heart of the pandemic, that we got together with our South African collaborators and thought, how do we do more? And here came a second shift or pivot in what we were doing. We decided that if we could build more research capacity, we could do much more. And that led to the receipt of a grant from the Doris Duke Charitable Foundation that allowed us to build the first dedicated biomedical research institute to ever be built at an African university. Certainly not something that I intended to do when I went to medical school, not something that I even intended to do when I started my medical training as an intern. That allowed us now to dig deeper into what was going on. And at the same time, there were others in the field that were working on this problem. And then in 2007, we had a real shock in that the vaccine that was being tested against HIV, which we had a lot of hope for, actually was a complete failure. And that really led all of us to re-examine what we were doing and how do we move the needle. And this actually, Dr. Fauci convened a bunch of us to talk about this. And what came out of that meeting was a sense that we really needed to do more to understand how the immune system works. So I myself started thinking, well, what is the answer here? And realized that we were not applying all of the scientific knowledge available to this problem. Because of the way that the scientific whole enterprise is built, it's built on grant funding. If you're not already in a field, you can't get into that field easily. It's siloed so that people in engineering aren't necessarily talking to the immunologists, and yet our belief was that those people had a lot to potentially offer. And that, in fact, there was no better place to try and build this kind of cross-disciplinary collaboration to take on a really major global pathogen than right here in Boston and Cambridge. And that led ultimately to the establishment of the Reagan Institute. The mission of the Reagan Institute was broad: to harness the immune response to prevent and cure human disease. Our initial goal was to take on the HIV epidemic and try and develop an effective HIV vaccine. Our strategy was cross-disciplinary collaboration coupled with flexible funding, and the location was really what I consider to be the very best biomedical research ecosystem that exists in the world. We were able at this point to dig much deeper, and I'm really pleased to say that just in the last month, we've been able to report that the identification of a particular kind of immune response mediated by killer cells that appears to be able to actually cure rare individuals of HIV infection. This gives us a blueprint for how to try and cure others who are HIV infected. And at the same time, so these killer cells basically will recognize an infected cell and kill it, as shown here. And at the same time, we've gone forward with our attempts to basically develop an effective HIV vaccine to prevent infection, and have what I consider to be a very promising trial underway in multiple countries in Africa right now, which will tell us whether we can actually prevent infection. All of that really happened because of the ability of us to pivot on a dime, to have flexible funding, to really take our ideas forward, and to have this kind of cross-disciplinary input to one of the biggest problems at that time that was facing our country and really facing the world. Now, in January of this year, I was teaching a course that I teach every year, which is called Evolution of an Epidemic. And here I am with my co-teacher, Howard Heller, and the 25 Harvard and MIT undergraduates that we take to each January for this course. And interestingly, two of the individuals who are on that course who I had invited are shown on this slide. The idea behind the course was to use HIV as a model to teach people how the medical community recognizes an epidemic, how you use clues from patients to understand what the cause of the pandemic is, and then how you develop vaccines and diagnostics and treatments, and how policy and advocacy influence that. Now, on the left here was a MIT undergraduate who had just returned from Wuhan. She started getting daily text messages from her parents telling her what a dire situation there was in Wuhan, with a new pneumonia of unclear cause and all of these people being hospitalized and dying. On the right hand here is a former trainee of mine, Diana Brainerd. We were visiting with a number of traditional healers, the traditional healers in the center there. Diana is now the director of emerging pathogens for Gilead, and had been overseeing the development of a drug there for Ebola. She was getting calls on a daily basis from people in China saying, can we get that drug, remdesivir, to try it against COVID-19 and see if it works. And it was the combination of those conversations that were being had with China that led us as a group to realize, oh my God, this is potentially another pandemic that's about to happen. So while we were teaching about HIV, we're suddenly pivoting to start talking about COVID. And it was on the way back from that course, thinking about what our mission and goal and strategy and location are for the Reagan Institute, that I realized we have to pivot again and start working on this pandemic. And that's exactly what we did. So what is this disease? It's a new disease. It causes a pneumonia, initially of unknown cause, but the viral etiology of that was soon determined. In fact, with unprecedented speed. You remember that it was 3 to 4 years before we had identification of HIV and a diagnostic test after the first public report of a new disease. Here it all happened in less than a month. Really an extraordinary accomplishment for the scientific community, built off of all of the efforts that have been done around HIV and other pathogens that help to build platforms to allow for this to happen. The virus that causes this disease is called SARS-CoV-2. It causes the disease COVID-19, and it's a small RNA virus. The genetic material is RNA. It has a spike protein that is the target for the immune response. And it was identification of this that led to rapid development of a diagnostic test. Now, what is a virus? Well, viruses are very different from bacteria. And let me just pause for a second to tell you about this. Bacteria are much, much larger, than viruses, as you can see here. Bacteria have thousands of genes, 4,000 in this particular bacterium, E. coli, and bacteria are capable of replicating on their own because they have enough genes to really direct all of that machinery. On the other hand, SARS-CoV-2, a typical virus, has 10 genes, and yet those 10 genes are able to commandeer the 20,000 human genes and force those genes to not make things for the body, but make things for the virus, namely new viral particles. And so it's really, I think that viruses are really an existential threat to humanity, and we really have to do everything we can to build the ability to fight these pathogens before they become pandemics. Where are we with COVID-19? Well, over 38 million individuals that have been infected. Fortunately, the case fatality rate is very low, but it's very transmissible. On the other hand, there have been two other pathogenic coronaviruses, SARS and MERS. Imagine, though, with these that had much less transmissibility, but much higher case fatality rates. I worry not just about the COVID-19 pandemic, but I worry about the next pandemic by a coronavirus where we may have the transmissibility of SARS-CoV-2 and the case fatality rate of MERS, which truly would be an existential threat to humanity. How does it cause disease? Well, this is a normal lung and a normal microscopic vision of what the air sacs look like. You can see these thin membranes so that air can be exchanged. On the other hand, look over here at what happens in SARS-CoV-2 infection, where you get this inflammatory response, all these immune cells that come in and basically infiltrate those air sacs and prevent oxygen exchange from happening. And what does the body do when one becomes infected? Well, the body has a way to target new pathogens like SARS-CoV-2 by antibody-producing B cells. When a B cell recognizes a virus, it starts to make lots of antibodies. And those antibodies are able to bind to the virus and neutralize it, thereby providing, ideally, protection from an ongoing infection. Now, the basic concept of immunity following infection is that you are uninfected, you get exposed to a virus, infected, your body, the virus spreads in your body, but your body's immune system starts to make antibodies and ideally clears the infection, and you're left with those antibodies, and you're immune, and ideally immune then from subsequent infection. The problem with COVID-19 is that the virus itself causes immune system damage as the virus replicates early on. And as a result, the antibody responses that are generated in this weakened immune system are at lower magnitude and are not as durable. And that's a very big concern in terms of the development of herd immunity from natural infection. On the other hand, think about what we're doing with the vaccine. What we're doing with the vaccine is trying to avoid the virus from doing damage by only showing the immune system part of the virus, in this case the spike protein. And what happens here is you take an uninfected individual, you immunize with the spike protein that generates antibodies, and you end up with immunity without ever having seen any damage from the virus. And now if you look in this hypothetical drawing here, the sense is that these antibodies will actually be longer lived because they're generated in the face of an immune response that has not been damaged by the viral infection itself. Now, in some individuals that become infected, they generate what we call broadly neutralizing antibodies, and there are now ways that scientists have developed largely around dealing with HIV infection to clone these antibodies and make large amounts of them, infuse them into an uninfected individual, and actually create immunity with the best antibodies available. So that's another approach. It's not vaccination, but it's something called passive immunotherapy. It's where you give a person, at least transiently, an immune response. And this is what President Trump was given. Eli Lilly and Regeneron and other companies are developing these, and they should be available, and I expect that they'll work very well. Well, where are we with a vaccine? There are over 200 vaccines for COVID-19 that are now in development, and these are the ones that have actually gone into human trials. We have a number that are in phase 3 trials. Some internationally have been given limited approval, but there are none that have been fully approved yet. We're hopeful, though, that that will happen soon. There are a number of different approaches. One is to just use the genetic material of the virus and put it either in a liposome or just give naked DNA where the genetic material from the virus has been spiked in. Another is to use an inactivated virus like adenovirus and splice in the genetic material so that the spike protein's made in the body by another virus, and that exposes the immune system. The tried and most used approach is viral protein plus adjuvant, and there are a number of approaches there. And then finally, inactivated viruses are being tried in a number of places internationally, but are not in development in the US. Our approach has been to use an inactivated adenoviral vaccine. This is basically taking a platform that is the basis for the vaccine for HIV that we have underway in a trial in Africa, and basically using that same platform to take some genetic material from SARS, put it in adenovirus, and see if we can induce protection. And that's been done by one of the founding members of the Reagan Institute, Dan Baruch. And basically, we started in early January. As soon as I got back from South Africa, the first phone call I made was to Dan. And by February 6th, we were already into animals to test that. And by mid-September, Dan already had that vaccine in collaboration with Johnson & Johnson in a phase 3 clinical trial. So really extraordinary that for a virus we didn't even know existed less than a year ago, we already have a vaccine in clinical trials. There are a number of other companies that are making vaccines now, and we'll have to ultimately decide what we're going to do in terms of prioritizing one versus another. Most of us hope that all of these will ultimately succeed, but there are a number of key questions that we have to think about. Does the vaccine protect from severe or only mild infection? How many doses are needed? There's 7 billion people in the world. If we need just one dose, that's 7 billion doses. If you need two, that's 14 billion doses, plus the additional logistic challenge of getting those people back for their second dose. How soon can the vaccine be widely available? Production is scaling up for all of these. Does it work regardless of age? And in particular, the elderly who are more susceptible, and also children for whom the vaccine will only really be tested once it's shown effective in adults. So we're a long ways away from that. Does the vaccine have to be kept in a cold chain, in other words, frozen? For the mRNA vaccines, that's the case. That adds a huge logistical problem globally. How durable is the immunity? Another big question. We're hopeful that it'll be durable. And again, how can safety be assured? I believe that the vaccines will be able to be shown effective given the processes that are put in place now to monitor them, and the fact that these have been in other people. This platform's already been tested in people. I think there's also a lot that we need to do. I'm really pleased that we were able to take the Reagan Institute sort of to another level by co-founding the Massachusetts Consortium for Pathogen Readiness, which I co-lead with Arlene Sharp. This is a collection of over 500 clinicians and scientists from across the Boston and Cambridge area that are all focused on sharing their ideas and collaborating to try and bring an end to this pandemic. So a few conclusions. One is that scientific advances build on prior knowledge, and the work that has been done on HIV and other pathogens has paved the way for us to respond rapidly to COVID-19. We have to be able to rapidly pivot, and vaccine development is now progressing at unprecedented speed, building off advances in HIV and other fields. I think vaccines will be available as early as late 2020, but they really won't be widely available until the second half of 2021. Multiple successful vaccines are going to be needed to meet global demand. So my hope is that all of these vaccine candidates will work. And in the meantime, I think we have to remember that there is a way to protect ourselves, and that is by wearing masks. The experiment has already been done. These are nurses in the intensive care unit at Mass General Hospital. They were up to their necks in patients with COVID-19, and yet by wearing masks appropriately, they did not become infected. So we know that masks work. In the meantime, we also have to think about what we as an institute can do, and I'm really pleased to say that we're now preparing for Reagan 2.0. This is a new building that's being built so that we can respond to future pandemics and so that we can understand the immune system in unprecedented detail to achieve our mission of harnessing the immune system to prevent and cure human disease. And finally, I'd like to thank the members of the Reagan Institute, an extraordinary group of passionate individuals who are fully committed to working on these important problems. And I'd like to thank our international collaborators as well that have provided so much in terms of our ability to gain traction. So with that, I want to thank Dr. Fauci again for the kind introduction. I want to thank InterSystems for inviting me to speak, and I want to thank you for listening. Thank you very much.
Developing a vaccine in the setting of a pandemic has been truly a surreal experience.
We've been working together really, on a day-to-day basis, maybe even hour-to-hour basis, trying to push this vaccine forward as fast as possible.
There's no magic bullet happening in the next month or so, but we're trying. We're trying very hard.
If we could understand what went right in those individuals that survived versus what went wrong in the individuals who did not survive, we thought we could put together a picture of the flavor of immunity that one would want to induce with vaccination in order to get protection from infection.
We largely used a playbook we had developed for HIV and then for Zika virus to develop a COVID-19 vaccine.
We certainly have a clearer picture of where we were a few months ago, but I still think we have a lot to learn.
What we know from the studies that have just been completed is that this vaccine is actually able to protect primates from becoming infected with COVID-19. What we don't know is whether this will work in humans.